Thank you. I'm the local application scientist for Labcyte, and fun fact, I got started using the Labcyte Echo in the Jeff Buca lab when it was at Johns Hopkins. For those of you unfamiliar with our flagship product, the Echo uses acoustic energy, which converges at the air-liquid interface. It forms Those sound waves converge, forms a mound of fluid, and from that mound of fluid flies 2.5 nanoliters or 25 nanoliters to an inverted destination plate, doing so with sub 10% accuracy and precision at that individual droplet size. So we can move any sample from any source to any destination well without ever touching it. Our proprietary technology, dynamic fluid analysis, is the key to successful aqueous transfers. This graphic illustrates how it works. The Echo sends a small ping of energy into the well. We capture these reflections as well as other fluid properties in order to generate a successful transfer. This technology allows the Echo to dispense any volume within range and can handle a wide variety of fluid types using wide-ranging calibrations that are turnkey ready to use. Since the acoustic transducer can move from any well, any source location underneath the well, and the destination stage can move independently, we can run many-to-one poolings. We can run one-to-many dispenses of precious reagents, and of course, any permutation that you desire using a CSV pick list. We now have several models of the Echo. The 550 and the 555 on the left are the instruments that generate a 2.5 nanoliter droplet. The major difference between the two is that the 555 is a bit faster. The 525 generates a 25 nanoliter droplet, is aqueous only, and therefore can build larger reactions faster since it's a larger droplet size. New for 2018 is the 600T series of instruments that now has the capability of dispensing 2.5 nanoliters out of a tube. And that is the tube, the fluidics tube that comes in a rack of 96. It was initially designed to be compatible for DMSO, for pharma, for storing drug libraries, but it would probably also do a good job holding DNA samples, yeast and the like, and share the tubes amongst the collaborators. In addition to ecosystems, Labcyte produces the Access automation platform, family of microplates, lids, and reservoirs for use on the ecosystems, and a suite of software applications designed to develop a number of applications. So now that I've showcased the products, now I'd like to share a couple of examples of workflows that are empowered by acoustic miniaturization. Probably the most widely used molecular biology application with the Echo is tipless, touchless PCR or qPCR. The Echo can be used to dispense master mix, cDNA, primers, and water backfills in a single protocol. This data from a 1536 light cycler highlights what we're able to do. We can dispense the master mix primers and the DNA sample to the green wells, and then of course dispense the master mix to the wells surrounding in the 1536. And what we can do is not amplify to the surrounding wells. We're not getting any crosstalk at 1536 well. Furthermore, we can generate qPCR curves that has a CV of sub 2% on the cycle threshold values. For the SynBio community, the following from our internal applications team highlights our ability to automate and validate assemblies. We've previously shown isothermal and Golden Gate assemblies in the sub-microliter range. Our in-house team recently assembled a 5-component assembly for a reporter construct using the AnyBuilder kit, and validated construction of that with a automated high-throughput colony qPCR method. So you can build DNA with in an automated high-throughput manner. You can also automate and parallelize transcription and translation, which can be showcased with the TXTL reaction. It's a great breadboard for prototyping gene circuits without interference from the organism. And it's particularly amenable to microscaling, as the GFP synthesis is oxygen dependent, and so the smaller volume, the greatest surface area that we can get for the reaction. To that end, we collaborated on an application note with Vincent Nero's lab, miniaturizing the reactions. And what you can see from the panel on the left, we can get a GFP readout from a reaction as low as 500 nanoliters. However, we're actually seeing maximal protein yield at a 2 microliter reaction. The figure on the right demonstrates a common theme with echoes in SynBio, and that essentially boils down to much faster execution times, and at the same time we're miniaturizing and doing it faster. Cheaper, faster, easier. Vincent's team also assembled a gene circuit with delayed feedback to investigate the response of the circuit, testing increasing concentrations of a promoter, of a repressor, holding the GFP reporter stoichiometry constant, and seeing similar levels of GFP readout when assembled by the echo as what's seen by hand. In the interests of time, I won't be able to go into the nitty-gritty details of the combining 2 promoters as well as a repressor with varying stoichiometries. But what you can notice is that we've triplicated 27 combinations rapidly. Lastly, I want to highlight a major value add in SynBio, one that in many cases has the echo paying for itself, and that is our ability to automate and miniaturize NGS workflows. We've brought Nextera down to running a reaction standard of 500 nanoliters, taking a $70 per sample prep down to 70 cents per prep. And our ability to miniaturize and reliably transfer in the nanoliter range has enabled our customers to not only see higher quality data, but also significant ROI by miniaturizing the various reactions. So I'll start to wrap up by providing a summary. So we're able to dispense 2.5 or 25 nanoliters with sub 10% accuracy and precision. We're not touching your sample at all, and we're not sonicating your sample. It's very gentle. You can transfer cells, beads with the technology. We can do any well to any well, so we have no fixed tips constraints. And as application scientist, I like to challenge that I can train any end user biologist to use an echo in about 30 to 45 minutes. No end user calibration, it's turnkey ready. I've only, of course, been able to scratch the surface on our applications in SynBio, so I'd welcome you to check out the website and see what we have in SynBio. And lastly, I'll wrap up with a familiar image for bioquantilism. Andy Warhol might not have recognized this image, but a clone picker just might. Thank you.
Good afternoon, everyone. Long day. So I'm Bill Peck. I'm the CTO of Twist Bioscience. And I don't know if everyone's familiar with Twist, but we're a 5-year-old startup. I was just reminiscing with Axel about Twist. Although for a startup, reminiscing is maybe not the right word. It's more like trauma therapy or something. But we are what we are. We're 5 years old. We've raised about $250 million for about over 250 people now. And so maybe it's a couple words for the organizers, and congratulations to the organizers of this, Jeff and George and Nancy and Andrew. You know, looking for funding and trying to find money and get people moving in the right direction is, the same direction, is always difficult. And when we first started pitching Twists a while ago, it was a different landscape, and it was hard to get money. And now we just closed another $50 million, and synthetic biology is the hottest thing in biotech right now. So things change. And I think your efforts here will be recognized, and we're proud to be founding, industry advisory board and a sponsor, and we look forward to great things in the future. And now I'm going to talk a little bit about what we can offer you in the future. So when we pitched the company, the idea was to bring a lot of the engineering that Bill Banier and I had into the world of synthetic biology. So we had a lot of experience in silicon and semiconductor and chemical engineering from our work at Complete Genomics, where Bill and I met. And of course, I'd worked with Emily for years at Agilent before that. So our idea was miniaturization, throughput, low cost, and make a versatile platform to supply many, many different product lines. And the stuff we're going to be working with you folks on, of course, is one of our major product lines, the gene product line. And this is, we're just echoing what's been talked about so many times today. I guess one of the things that was so motivating and keeps me interested in this project is coming from an oil country. I come from a place in Canada, Alberta, Canada, which is famous for its oil tar sands. And actually, before I got into biotech, I was in fluid mechanics, and I did the air quality modeling for what the oil sands were going to do to get all that oil out of the ground. And we're going to destroy the boreal forest, but all in one go. So we've had a good chance at that. But this type of thing is really encouraging to me to think what we can do in the future with a lot of the work from people like you. So knucklehead engineers like me can actually contribute to something useful. So you think of the products made by oil now, maybe in the future we're going to be using organisms and modified organisms to provide the same quality of life, and but at a much less impact on the environment. As same thing. We've talked about this in crops and all the idea that the way we can modify crops, and I'm taking the gentleman's comments on GMO to heart, and we're very interested to hear more about his idea on how to avoid that pitfall. Another thing is, although global warming seems to have been solved with the current administration, when it comes back, we have to think about how to respond to climate change quickly and modify our crops quickly to work with the new climates. And of course, healthcare. It's a big product line. We offer lots of like NGS solutions to gene solutions to a lot of researchers. This, of course, is immunotherapy. So our technology, what were we up to? We're up to trying to deliver genes quickly and with high quality and high throughput. And that was sort of the reason Twist was formed, was we realized that to get the experiments done on scales like you're talking about, that requires massive, massive throughput. So what we were finding in the market research was scientists were artificially constraining the size of their experiments based on what was available. Makes sense, right? So how do you expand that size? How do you elasticize that market? And we thought, well, you take the traditional way, 96 oligos are made in a 96 well plate, you get a mermaid or something like that, and you combine them and make a gene. We just took our experience with silicon and said, well, if you miniaturize that into using all the beautiful abilities that silicon give you, all the control and engineering control that silicon gives you, you can think about doing that on a scale 10,000 times greater, which is what we're doing now. So what can Twist do for you? As I said, our major product line is genes. We also provide oligo pools, just straight up oligos. The PEGS conference is in town right now, so I'll be going there quite soon after this meeting to talk with the library group. We have an NGS solution, next generation sequencing solution, and data storage. And so I've been sort of leading the data storage charge, and every time we talk about data storage, I get the same question: is it real? And Victor Zhernov is here, who's been a great champion of that through the SRC, the Semiconductor Research Corporation. And it is real, and for many different reasons. And it's on a scale vastly more massive than anything we're thinking about here. The subtleties aren't nearly as complex as the molecular biology. And it's already real. And what it's done already, when we talk to investors or the press or whatever, it articulates your message, that the biology of information, right? So information's biology. Biology is information. And people with data storage, they get that immediately when you talk about how the analog between the information stored in a genome and the information stored digitally. So it's already proved very, very useful for you. Synthetic genes. All right, so here's your gene, your vector. This is the products we offer already. We can support the antibody-based drug development, so it's a broader platform. Gene editing, we do the donor DNA synthesis, gene therapies, and pathway assemblies. Now here's I see many of our early customers, and it was a rocky start, but we're scaling, and the scale is starting to work. That ramp was not constrained so much by hardware, more by software, okay? So it's been a great learning experience on how to deliver a product. It's hard, but it's getting much, much better. So I think last month we delivered 29,000 genes or something like that. And the first couple months we're struggling to deliver 29. I'm not sure what that is. So, and of course, one of our great partners has been Ginkgo. It's been, if you've followed us at all, we've had this partnership with Ginkgo, a Boston company here, who are an application course company. We're the tool provider to that application, and one of the important things if you do start a startup is to find and you go commercial, is to provide a properly sized complement to your product. And Ginkgo is the perfect complement to our product. And it's a 1 billion base pairs of DNA delivery. I mean, when we first started the company, it was just unimaginable that we could deliver that or that there would be a customer needing that type of, well, it wasn't, we imagined it, but to convince investors that it was real, it was unimaginable to the investors, and now it is. Rather enormous inverted triangle, but it just gives you an idea of when we kicked off the company. It's in SynBio Beta in 2015. I don't know if John's still here. John Cumberson is in SynBio Beta. We've gone up 10x, right, the number of base pairs we're synthesizing. And we also offer online ordering. So anyone who's coming to our website now, you can do a drag and drop and order online, sort of the e-commerce or integrated ordering system. Thanks a lot to Omri's old team at Genome Compiler. They've been doing amazing work on getting this up and running. And off we go. So this shows you an ordering process in real time if you really know what you're up to. You design it, we build it. And then, so I am the CTO, but I do get to talk about pricing. So right now, it's gene fragments are 7 cents a base pair, so those are non-clonal, and clonal genes are 9 cents a base pair, 20 business days. But for the special volume discount for the GP Write people, and we can talk about this in more detail later, but we've reduced the price substantially, 2.5 cents, and then 5.5 cents for clonal. And we can talk about aggregating orders and finding out what works for the members of the consortium in supporting you. So that's all I have to say. A couple of minutes left for questions. Glad to take any questions.
Hi everyone, my name is Helge Bastian. I'm with Thermo Fisher Scientific and run their synthetic biology business. Glad to be here. Thanks to Nancy and the organization GP Write to have us again, the 3rd year in a row. And how amazing this is, right? Unfortunately, I couldn't attend the whole meeting, but I can tell you, you all are in really good company here because this is, somebody said it before, the most exciting field in biology right now. And I don't know if you discussed this during the day, $1.7 billion have been invested for our startup companies in SynBio, outpacing the whole industry as far as startup funding. So it's tremendous. And it was, because it's so tremendous, other organizations that I visited yesterday, the ALDA organization, which is a life science organization since 1988, not so much specialized on SynBio, have put together a senior executive spring meeting to focus on SynBio, to educate themselves on SynBio, because it is just so exciting and has tremendous potential, not just for the research market, but beyond, right? So we all know this. So Thermo Fisher, as you may know, it's around a little longer. We consider ourselves the leader in serving science, and that's exactly what we're trying to accomplish. So we are not a synbio company per se, we are a tool provider, and that's our supporting our mission. We try to enable our customers or companies that are in the area to be successful in what they're doing. So when it comes to the synbio initiative in Thermo Fisher, we're trying to help the synbio community to just get along much further and faster, and this is where we develop the tools. So if you look at our portfolio, I can jump to the next one. This is essentially what we consider our portfolio in synthetic biology. So we provide everything as far as the building blocks for making DNA and RNA. We do make DNA and RNA, and also we do make DNA and RNA not just in silico and in vitro, but also within the cell when we apply genome editing technologies and develop gene editing technologies, or on behalf of customers also do the cell engineering piece, right? And this is what I'd like to lead you through a little bit, what have been done in the last year, actually, because we were here last year already. What have we done since, and what are the tools that we can make available for you guys? Don't want to be too commercial. I just really want to be informative what our R&D does and spits out to help our customers to be more successful. So if people ask me what are we really doing, the simple summary is we are in the business of engineering nucleic acids, because that's really what we do, right? That's the deep expertise within Thermo Fisher, and we're trying to figure out what you guys need and try to provide that. And then I found a very simple definition for synthetic biology that people have. It's essentially engineering biology, right? And that's what we are all interested in. And interestingly, that goes beyond the life science community, right? Because it's like with the 23andMe in the US, it's like all of a sudden the consumer is involved, right? So the many application that we saw in other slides is touching the consumer. And this is what it makes so interesting for life sciences, right? So again, this is what we're doing too, and I think it resonates a lot with our employees and hopefully with our customer base too. That a life science company that has an initiative to really fully understand how to design and make a nucleic acid, because it defines life science, and at the same time, the direction is to go into data science and digital science, develop a digital science company. And how more digital can you be if you understand that the DNA essentially is a digital media that biology is run on, right? So can't get any better for biologists. So what have we done in the area? We mentioned this last year that we're on the making progress in developing the libraries, the whole genome libraries. This is done now. So for those that do knockout and screening procedures, this tool is available. It's a major product for us because, as you can imagine, it's a freezer full of tubes. We can get it in lentiviral format or otherwise, and it's available, right? And then we have arrayed libraries, so you can pool if you want, but you don't have to. And for those that are in the screening business, it's very helpful. The other advancements we did is when it comes to further improving the tools. Well, that's what we do as a tool provider. So the latest one is that we brought up a new Cas9 enzyme that increased efficiency significantly. And since the application are so close to the clinic, we also test in relevant cell lines, right? So something I want to bring to your mind if you haven't seen that already. If we move on, the other efficiency that we try to improve, major interest in genome editing is in the knock-in applications, especially when it comes to precision knock-ins. And the way we approach this is to optimize the reagent formulation that allows us to not just only bring the active reagents into the nucleus where it's supposed to be, but also to make sure that the whole organism or cell metabolism is helping us to do the editing event, right? Last but almost not least in genome editing is our precision approach for knock-ins. And as you may remember from my talk last year, CRISPR is of course working so nicely, right? But still there's areas in the genome that you cannot reach, especially if it's a PAM-dependent CRISPR, right? So this is where we have revitalized the telefactors, and the telefactors are very, very efficient and can reach essentially everywhere in the genome. And since this works so well in our hands and is a complementary technology to CRISPR, we also opened an early access program for those that are interested in this. And you can call us anytime. We're happy to, if you can't reach your target in the genome with CRISPR, we're happy to help you with the telefactors, right? So the update on our DNA synthesis technology, we have a slightly different approach than what you heard just from Twist, right? Because we don't want to consider this just a manufacturing technology, but also something that we can consider goes back to the bench, where maybe some of you or others are interested to make genes and oligos yourself, which is not necessarily how it works today. So what we have developed is a new tool that is based on a chip that works electrochemically. Here we're talking about 35,000 oligos that we can make at once at very small quantities. And the quantities you see on this one is about 100 femtomole per oligo. Plenty of material for those that are in the gene synthesis area. So we don't need nanomole, we don't need anything major than femtomole is usually good enough, at least in our hands. And so this is the next development that we have. And what we try to do from here is to bring it one level further. And but what we do in the meantime is we test applications with people that are not necessarily in the science industry or life scientists, but have specific problems to solve. So in this one, I don't know if you heard about this, it's a project that we call Game Over for Aflatoxin, where we were approached by the Mars company, the candy bar company, that is really fighting with the issue of aflatoxin that is around for as long as I'm on this planet. And now they're trying to apply synthetic biology approaches to find a way to get rid of the aflatoxin, which is a major issue because it causes cancer and other not so nice things, and they need to be tackled, right? And the approach here is through gene synthesis to find ways and enzymes that can help digest aflatoxin eventually, right? So one application where we apply our new technology, but another application that we're thinking about, and it really depends a lot on the folks in the room, but also other customers in the area, whether it's the time to bring gene synthesis really back to the bench, right? We are a service provider too, there's many other companies here, but the big question that we have is, if we make it easy, if we make it less cumbersome, and if we make it not smelling like organic chemistry, there may be an interest to bring it back to the bench. We can make a lot of oligos in a very short period of time now. We can make it at very tiny amounts, and Labcyte may be interested in the workflow here too. But we also want to put the assembly back into the lab for those that are interested, right? So therefore, this is the next, what hopefully I can talk about next year, that this is something that would be available for you too. And in the meantime, we're always looking forward to partner and collaborate for those that think there's an interest in technology, not just on the commercial side, but also as far as collaborations. I think that's my last slide. The rest is a summary, if I'm remembered correctly. It doesn't even move. Yeah, the rest is a summary, and I open up for questions if we have time.
Yeah, very interesting new platform. Can you give us some idea of what you think the per base consumable cost will be on the new platform, and also the instrument cost?
Yeah, so well, we don't sell the instrument today, but the instrument is probably in the neighborhood of what we heard from the Labcyte. Completely different technology, but that's probably what you have to play with. When it comes to per base, I would answer this way: price is not really the issue. So what we're hearing from other platforms, I think the industry is looking for a more efficient way to do lots amounts of nucleic acid. So we're more betting on also the convenience when it comes to a modular system that is not just sitting somewhere in a warehouse and producing for others who don't know what it is, while we try to bring it back, as I said, to the customer. But you will enjoy the same pricing as the whole industry has to deal with, right? And we have seen this over the years, that the price for DNA, of course, came down significantly. This is why these platforms have been developed, so that everybody can be competitive in this area. Nancy, you had a question?
Yeah, so that was really interesting to me, and I'm wondering if by trying to bring synthesis back to the bench, you're actually thinking about another epic battle between companies like Illumina and Life Technologies had over the sequencing market,
Absolutely.
which really helped to drive the technology development and the price down. Are you seeing something like that emerging?
That's a very nice analogy, because that's exactly what happened, right? I mean, when we look back for normal oligosynthesis, this, of course, there were oligosynthesizers 20 years ago, then it turned into a made-to-order kind of industry. And the sequencing, the same thing, right? There were sequencing labs, and until Illumina came up with something that makes it really efficient, and now Thermo Fisher luckily too. I can see that indeed, right? And so because there's many people that are very impatient to wait for the ready cloned or linear gene, so why not doing it themselves? If it is idiot-proven, right? So the idiot-proven part should be for the oligo manufacturing itself. And I heard from the foundry before, you don't make oligos, maybe it's not so time-consuming that you may think. And of course, the assembly need to be idiot-proven too. But the industry has shown that many of the enzymatic reactions, be it PCR or whatnot, or sample prep even, right, it can be put in a box. It might be not welcomed by most of the professors that teach life science, but for the industry and for moving faster, I think it's probably the right thing to do.
So can the synthesis service providers coexist alongside that?
I think so too, yeah. I mean, people and companies have different objectives, right? The public companies are always under pressure to make sure that they make their top line and bottom line numbers. And as a private company, you don't necessarily have this pressure, and people may be able to live with lower margins, right? So if the service industry usually, as everybody knows, a lower margin than in the product industry, that's just the nature of what it is. So I think it can serve everyone, right? It can serve the industry, it can serve the research, and it can serve those that are interested in running their own business. There's a lot of smaller companies that like to do this. They buy oligosynthesizers today, but there might be time now to bring more recent technology on the market.
So for the oligosynthesis platform, can you tell us something about the error rates of that synthetic material, particularly as it relates to the desire to build larger genes from that?
Yeah, absolutely. And so we have different platforms, right? And all of the platforms we are using for our gene synthesis, we haven't taken to the market in the past. That may change. And depending on the platform, the error rates are also different, right? So we have everything between 300 and 700 per base. And at the end of the day, it also depends on your downstream process, so how you stitch the genes together, how you do the error correction, and so forth. So, but it's between the 300 and 700, depending on which platform we are using. And for those people that don't know, sometimes it's an open secret in the industry, but make no mistake that the oligosynthesizers that are used for making primers for PCR are, of course, different when it comes to error rate specifically and quality of oligos than what you would use in a normal PCR experiment, because this is where it matters, right? So, of course, the folks in the industry know this. Sometimes I realize customers don't, and they order oligos because it's an oligo, but they don't specify for what application they're trying to use it, right? And sometimes it's useful information if they order from at least companies like us, because you can choose from a number of platforms, right? Thank you.
Hi, folks. Nancy thought that folks would be interested in hearing about one of the recent developments from the SBAL community, which is about how to better show diagrams. And I'm really hoping that everybody in this room is about this close to using this already, because the sort of diagrams that we're talking about are essentially boiled out of the emerging conventions of how we all have been moving towards drawing diagrams in the papers we publish and the all the tech notes and such that we put out anyway. So what the SBAL community has been doing for its diagram language is essentially looking at what everybody's doing and what everybody's trying to talk about in their diagrams and trying to boil down those conventions into one consistent, well-backed semantically language for our visual diagrams. So in an SBAL visual diagram, you have everything's kind of based around the nucleic acid backbone, from which you have sequence features shown on that with glyphs. You can have other non-nucleic acid species as well, like this GFP molecule being produced here. Interactions like genetic production, repression, and of course names and labels and annotations on all of this as well. So hopefully, this is all not too far from the sort of diagrams that you draw already. And we want to basically encourage you to look at this standard and be a little bit more intentional about making sure that you use the same sort of glyphs and the same sort of rules for clear communication so that all of us can understand what you're doing better and vice versa. There's a lot of flexibility. The SBAL visual approach has tried to confine things as little as possible. So think of this as more like a specification of an alphabet, but you get to pick the font and do what makes most sense for your artistic and diagrammatic needs. So color, text, how you scale things, whether you're showing one or two strands, lots of stylistic choices are all specifically not constrained by the diagram standard. So everything that's up here is legitimate SBAL visual produced by different people, applications, scribbles on the whiteboard, etc. Here's another example of a somewhat more complex diagram with two functional units, some parts labeled, some parts not. The top one producing TED-R that's repressing production of GFP on the bottom one, and we've got some more species there. These TED-R and GFP here are being shown more properly as macromolecules, and we're, in fact, in the bottom part of the diagram, we're explicitly showing the intermediate mRNA stage and degradation of those two. So, level of detail up to you, what you want to communicate in the diagram. So at this point, in addition to the sort of base language, there's also a fairly large collection of glyphs that have been developed and are continuing to be proposed and developed by the community. It's an open community. Anybody can come in with new needs and propose things. So here are many of the nucleic acid glyphs, coding sequences, restriction sites, origin of replication, etc. Sometimes there are recommended and alternative ones when there are two conventions that we need to support by long community history. And a whole collection of these. In addition to the nucleic acid sequence feature glyphs, there's also glyphs for molecular species, in many of the main categories that people tend to be putting on their diagrams, and for major classes of interaction. I'm also happy to announce that actually right now we are in the middle of the adoption vote for new symbols for denoting genomic context, motivated by discussion of a number of projects, including this one. A new symbol for circular plasmid so that you can designate that a structure is circular, or for chromosomal locus so you can talk about integrations. In fact, the proposals for these, the vote is running right now and it looks like they're going to be adopted. So we'll be able to talk about genomes in our diagrams in an official way very soon. And the ontologies this is associated with have had the their appropriate extensions as needed too. So if you want to start doing this, do it by hand. Use your favorite graphics editor. Most of this can be done directly. There's a glyph set available online at the SBAL site. There are some specialized visualization tools that people have put together also, and there's even, we also just pulled in a community-contributed font, which has a user-defined symbol set that has all of these glyphs in it. Great work by a guy out of TU Munich. And I'll just end by, first of all, encouraging everybody to use these symbols in your papers and in your talks. Every engineering community ultimately lands on a set of diagram symbols that are useful for it. The mechanical engineers have it, the electrical engineers have it, the architects have it, plumbers have it. We should have something that we're agreed on too. And since the diagrams you're already drawing are probably about this close from it, I encourage you just take that one extra step. Second, where it's awkward for you, or where you want to express something that there's not a good glyph for yet, or where you have questions, go to the SBAL website, go to the community pages for this, go to the GitHub site where all of this is put up, and raise issues, contribute opinions, add use cases, add the symbols you need. That's why we've got new genomic symbols coming in. This is an open community. Everyone is invited to participate. Anyone's encouraged to join and help curate. And the way that this will evolve to best serve all of us in this room and our whole community is if you make your opinions known and make sure that this is all serving your needs as best it can. With that, I'll thank you, and we may have time for a couple questions. Thank you. I see Pete back there has a question.
Well, so Jake, you've already touched on this briefly, but kind of presentation in a good way, begging the question for more of, this is for both you in the room, is what future needs in this category does GP Write seem to evoke? What unmet need might we be thinking about right now?
I have a suspicion that at some point we're probably want to be a little bit more systematic about how we draw diagrams about chromosomes. There's some sort of loose conventions, but when we were working on these genomic context glyphs that I was just showing, it became very clear that there's not really much agreement on, how do I draw a chromosome? How do I draw a locus? What is a locus? There's some conventions, but I think that's an open direction. There's also some things that people have kind of bounced off of a couple times and been like, maybe we're ready for that, maybe we're not. What's the right way to draw? For example, a hammerhead ribozyme. Does anybody care about that enough to standardize it yet? It hasn't happened yet, but as soon as there's a few users, in our community who are finding that a convention would be useful to them to make their diagrams clearer, it's very easy to go there.
I would encourage the community that this is great and is absolutely necessary, and I would encourage everyone to not let the best and the perfect be the enemy of the really, really useful. And this comes from experience dealing with genetic variation. They are still haggling on the BCS format for really deviant points of mutation and difference, and it's incredibly useful to have a standard. So if this can't do everything, we'll get 95% of the way and complain about it.
It's also true, one of the official things that's in the standard are rules for how to make up your own glyphs. And all you basically have to do is, you can make the green eggs and ham glyph and all the on beyond zebra glyphs that you want. You just have to make sure that you aren't drawing something that looks like a promoter and telling people that that's not a promoter. All right. Any last questions? Yeah. So this is an open public community. Anybody who signs up on the mailing list and pays attention has a vote. And in practice, what happens, the way that this actually works out. So like I said, we just went through this process with these genomic context symbols. And what happens is that people who are invested in it end up having a long discussion. Once there's consensus among all the people participating that this seems to be good, then the rest of the community gets bugged. Hey, wake up, take a look. We think we may have consensus on these symbols. We think they work, and here's why. There's a whole form for writing out why you think this is good, and what works, what doesn't. What were the alternatives that you decided were not the right way to go? You wake up the rest of the community, and then people say, oh, hey, I should have been paying attention, and I disagree with everything you wrote. And then you go through the consensus process again, only usually a little bit faster because you've already, ruled out, you've already got good reasons to not do a lot of the alternatives. But things may change a bit. And eventually, when you've got consensus enough, it moves forward to a vote. So anybody who wants to be involved is strongly invited to participate. Because this is the legitimacy of this is simply open to the community. Whoever participates, and nothing goes forward without near complete consensus. All right, thank you very much.
Hi, my name is Ybert. I'm the CEO and co-founder of DNAscript. So we presented our company in this meeting 1 year ago, and we are happy to be here again this year, thanks to the invitation of the GP Writes conference organizer. So we are a startup company backed by leading industrial and financial investors in the field of genomic and life science. We are based in Paris, France, and we have today a team of 27 people, mostly in R&D. We design, imagine, and develop our technology mainly ourselves, but we also have key collaboration, one of them being with Institut Pasteur since 2014 when we started this journey. So the reason why we have started this journey is that our goal is to unlock the power of DNA to write the future of life science. So our common objective today is to achieve synthesis of whole genome. In other words, being able to synthesize extremely long fragments of DNA with an absolute precision. This process involves the synthesis of a very large number of oligos and then carefully and patiently assemble them into longer and longer constructs. We think that this whole process could be improved. And it could be improved by using longer oligos and by using oligos with higher quality, higher purity. So longer oligos are indeed simplifying the overall process of assembly and increase the reachable length of the final construct in a given amount of time and work. Purest oligo decrease dramatically the need for verification and the need for restarting the synthesis work when there is a failure or an issue with some oligos used in the assembly. So having access to very long and very pure oligos can significantly change our ability to synthesize whole genomes. And today, the length and the quality of the oligo that you use totally depends on the way that they are synthesized. So historically, people have used different variations of the same general techniques to synthesize oligo, using organic chemistry to achieve sequential synthesis of the DNA polymer. And the key metric of this process is the coupling efficiency. It governs the maximum length and the overall quality of the oligo that is synthesized. And the latest improvement on the chemical synthesis of DNA has been introduced in 1983 with the description of the phosphoraldehyde method. And this method is still used today at every DNA provider factory. We believe it will be difficult for the chemical approach to go far from being the current limit of 99.5% coupling efficiency. That means that the maximum length and purity of your oligo will remain limited. This is why our approach is radically different. We deeply engineer a polymerase enzyme and we perform nucleotide chemistry in order to develop an enzymatic-based DNA synthesis. So polymerases have evolved during billions of years to be super efficient at coupling incoming nucleotides to a growing chain of DNA. So by using them, we get access to an unprecedented level of performances regarding the coupling efficiency. We also get access to super fast coupling reaction and to a DNA-friendly environment without any solvent or any harsh chemical. Everything is happening in water. So last year, we came here to present you our project of developing this technology, and this year we are here to show you some results and to give you an R&D update. So first, the performances trajectory. So we have been developing our technology at a very fast pace, and we plot it against the phosphoramidite development. We expect to follow this trend for the following year to come. Of course, as it is the case with any disruptive technology, we need additional time to reach the maximum level of performances. But from our current trend of results, we are already expected to be above the phosphoramidite performances in less than 1 year. So what does that mean particularly? What are we able to synthesize today? So today we are able to synthesize de novo fragment of 50 nucleotides in routine. So we do it in an automated way using a different type of hardware, and we can parallelize the synthesis. Our efficiency per cycle is more than 99%, and you can see one of our typical results on this slide. So this is a crude oligo. There is no purification. It's directly from the tube into the gel. So this is this part. And as I told you earlier, our goal is to help you, not to set technological records for the sake of doing it. So this is why we wanted to test our oligo into real condition. So here we decided to synthesize 2 different primers, so T7 forward and T7 reverse primers, and to use them into PCR amplification experiments with the goal of amplifying a 373 base per fragment between the 2 primers. So first, we synthesize the 2 primers, and you can see the synthesis results on the left part of the slide. And then we perform the comparative PCR by using our oligo enzymatically synthesized and compare them to to oligos from traditional provider as a positive control. So as you can see, we were able to successfully amplify the sequence with the 2 primers made enzymatically, and the band has the same side and the same intensity compared to the positive control, where the same primer has been coming from chemically synthesis method. So we believe that this was the first time a PCR experiment has been done with fully synthetic primer made from de novo enzymatic DNA synthesis. So what's next? We are confident we will raise the level of performances of this new technology in the coming weeks and months. Our team is making very good progress on the different aspects of the technology in order to reach unprecedented level of performances for DNA synthesis in 2020. So this is particularly true for the length, the purity, and the time of synthesis, and of course by keeping the process very simple without using any organic solvent to have a positive impact on the cost. So to conclude, we are continuously making progress on our technology. What you have seen today is just a milestone result. More will come, but it shows that there is a real alternative for chemical synthesis, and this is emerging. Based on our progress, we believe that this alternative will soon overperform from traditional method. And to wrap up this talk, I would like to invite you to think about the possibilities that you will have using this new technique, and so getting access to very long and very pure oligos, to being able to get access to your DNA super quickly, to have access to modifications such as control epigenetic DNA, for example. This is one of the modifications we look into. And to even synthesize your DNA directly on your lab with a very simple process. So think about it. What will you do with it? So if you are interested in being involved, please come to find me after the talks, or contact us by email. Thank you very much.
Yeah, hi. Thanks for the talk, very nice. So how does your approach differ from other companies that try the enzymatic approach?
So in our case, what we do is we really deeply engineer the polymerase so it could fit into the requirement for the performances of the technology. So we don't compromise over trying to use, for example, easy to apply chemistry on the nucleotide. We choose carefully all the chemistry so it can bring the full potential of the performance, and then we deeply engineer the polymerase to be compatible with it. Thank you. Okay.
So you're deeply engineering a DNA polymerase, which usually takes a template. So you're saying that you have a template that it doesn't copy, correct?
So we use polymerase that doesn't need a template. So we use many different polymerase, and the common characteristic of this polymerase is that they don't use any template to be active, and we start with that, and we continue to reinforce these characteristics. So as I told, so today our overall coupling, our overall efficiency per cycle is more than 99%. So the efficiency or error rate will be similar to a traditional chemical method, which is governed by the coupling efficiency or the efficiency per cycle. Okay, thank you very much.
Well, good afternoon. I would like to first thank the organizers, like Nancy, for inviting us to this conference. I'm going to speak about gene editing, and the idea is, so we're a listed company, you have to read this. It means everything I'm going to say is like just your responsibility, but you can read this later. I'm going to speak a bit about the history of gene editing, and I think the history of gene editing has been very much imprinted in this area, and I would like also to share a bit of what I think has been constructed over the years. So a bit of this history, and it goes from like homologous recombination to nucleases. And in 1983, I think that was probably one of the founding experiments by paper published by Jack Szostak and colleagues here around that showed that breaking DNA with another DNA that share homology induces what's called a double-strand break repair. And this double-strand break repair will use homology upstream and downstream this break and will lead to a gene conversion. A gene conversion at the, well, have difficulties to find a laser, at the place of the break, and insertion of some genetic information at the place of the break. Interestingly, when you make a break here, the expansion of this gene conversion will have a bell curve. And the speed of expansion of this is also conducted by the composition of DNA behind this. After this experiment, then essentially one researcher has been constructing a lot of the knowledge around gene editing. I think that's probably the experiment that were the founding experiment of gene editing and the use of this double-strand break repair and homologous recombination to tweak DNA. Everything has been done in yeast up to now. And it's like Rodney Rothstein, professor at the University of Columbia, that has described that you can make recombination either like by integration with end-ins or end-out. Currently, everything that is used is end-out homologous recombination, inserting some markers in there. 1985, that like enzyme called omega, or ISC1, that the name came after, has been cloned by Bernard Dijon. I think that this enzyme was cloned in George's lab, as I remember. Bernard was my advisor
So, like, yeah, as I can recall, he was making these experiments. He cloned this enzyme, like, right to the bench close to George. And that's probably the old enzyme that started, like, the ancestry of gene editors. And what makes it a gene editor? It's cryptology. It recognizes over 18 base pair. It means like 4 to the 18th, which is 6.8, 10 to the 10th. It's probably like more than 10 times over the human genome. So you can use this enzyme and cleave DNA inside living cells and edit genes. And in 1998, an experiment was conducted that was gene replacement by homologous recombination in mammalian cells, where upstream homology, downstream homology, and a sequence between the two that replaces a sequence of the HOX3 gene that was conducted by Lemoylik and Brûlé. And that experiment has been conducted, and it's probably one of the founding experiments in mammalian cells, followed by combination of all this stuff that is combining a nuclease on one side to break DNA and using this N-out donors thing where you can insert sequences inside DNA with a pretty high efficiency, because natural homologous recombination is fairly inefficient. It works in 1 time over a million. You can raise this efficiency, but if you cut DNA, this would be way more powerful. And like that's probably one of the main first patent that was filed 25 years ago in May 1992, 26 years ago, by the way, 92. And let me describe this, like a lot of crazy ideas, like how end joining would happen after like making homologous recombination, like a DNA cleavage or loss of heterozygosity, and a lot of different things. But also in 92, a lot of description on how we can delete a gene, insert a gene, replace, even super crazy things that, like that in 1992 were like discovered, or described at this time in these kind of patents, where you can delete totally loci with a full set of genes around here. And I think this has not been done yet by nucleases, but it was described at this time. And also, like the idea and the goal was essentially to try to do this for like gene therapy and including immunomodulation with interleukin genes. And I'm going to end up my slideshow by showing some experiment using interleukin genes 26 years after. So talking by gene editing, it's essentially like you can edit cells for therapeutic application, either to destroy tissues or regenerate. I'm going to accelerate because, like as I said, it's a bit off topic. And it's interesting to work on cells, and that's why we're super excited in being trying to contribute to this effort, the Genome Writes project. And I'd like to thank you for this, because I think that's probably one of the milestones in human history that is conducted here. And I'm extremely proud to be able to participate to this, because I can go to Mars or to the moon or put a lot of different things. What you're doing in this Genome Writes project, I think, is going totally to change life on Earth, and it's going to be really interesting. It's a turn in history. We're discussing about we were talking about the ethics around this. I think it's like a really huge subject, and it's mind-blowing, but definitely one of the first key applications will be how to use the cells to treat people and to cure people, and that's what is the main interest we have in Cellectis. So the real challenge behind this is trying to find a site that is as long as possible. So size does matter with nucleases. The longer the nuclease recognition site is, the better it is. If you recognize 3 base pairs, it's like 64. If you recognize 32 like a talon, then you're at 10 to the 19th, even though if in the recognition, it's not 100% full around this. And I'm going to describe a bit of this. So currently you have 4 families of nucleases that can recognize more than 18 base pair, which are meganucleases discovered in 85, zinc finger nucleases essentially set up in 2003, Tal nucleases, it's 2010, and CRISPR-Cas nucleases in 2012. After this, you can combine them, or there's other members of the family around this. For example, meganucleases, I think the family is like over 800 different members. Talon, there's 2, 3, 4 different members, etc. Cas also. So it's not limited, but you can combine them. For example, you can combine tau nucleases with meganucleases. It makes megatals, etc. Boom. So why we're using TALEN? TALEN is interesting for a certain number of reasons. It's composed of RVDs that recognize each of them a base pair. And each of this RVD recognize A, C, T, G, and you can place the cut almost at the base pair. I think the most distant place compared to the place where you want to place the cut is 6 base pairs, and that makes it the most precise. You don't have constraints as you can have with sometimes 1 base pair or PAM sequence, and that's what makes it fairly strong. And also one interesting thing, it has been in the clinic since 2015 and approved by the FDA to go in patient, and I think that's probably one of the most powerful take about like TALEN. When I was talking about RVDs, so you don't have one RVD per base pair. We have a collection that more than 400 RVDs. Each of these RVDs can recognize have more than like 100 RVD recognizing A, more than 100 RVD recognizing C, etc. And you can always reshape the enzyme up to the time you can totally suppress an off-target site. So there are, for example, what are called non-canonical RVDs that you can switch between one and another to totally suppress an off-target cleavage site. And I think this is one of the most powerful things where you can re-engineer an enzyme behind this. Building a TALEN is pretty simple. You just do this very simply with using Golden Gate technology, and it takes 5 days to do this. It's quicker than synthesizing an oligo. And the effort is not that much an effort in building the enzyme. It's essentially a computational effort in trying to make the best assembly to make the best TALEN, and the success rate is extremely high. It means every time you make a TALEN, you can always cleave the site. After this, you can improve the TALEN, but you always have something that would induce a cleavage. And here is, like, to wrap up this, is like the idea of using this type of technology in a synthetic biology fashion. And that's done in T cells. Let me give you an example here where you can making a double-strand break with a TALEN at the TCR alpha locus, and you can insert here a CAR gene at this site. But on the other side, you will use another TALEN and insert the interleukin 12 just after the CD52 gene, after a 2A sequence. And so you will suppress the expression of TCR alpha, and you will keep the expression of CD25, driving the expression of IL-12. Why IL-12? IL-12 is a very powerful anti-leukemic agent, but the problem is that you cannot dispense IL-12 in the blood, like systemically. If you dispense it systemically, it will be extremely toxic in the patient. So it's not great to dispense this. The best thing is to dispense IL-12 right on the tumor. And we're going to use and resynthesize a bit of like real small portions, like just the beginning of what can be done. But redesigning the T cell in order to make the T cell like a spray, and the T cell to spray IL-12 right on the tumor when you challenge the tumor with a T cell. You see here, that's like the T cell, and that's the expression of CD25. You put some tumor with the CAR T cell, and you will see that IL-12 will be expressed, and once all the tumor will be killed, it will go down. And you put again some tumor, challenge again, whoops, it starts again and go down. And you see that if you follow up the expression of IL-12, that is just after CD25 here, you'll see that IL-12 is expressed and down after the tumor is killed, or again expressed here. And you see what kind of effect you can have on a tumor. That's the tumor alone. You'll see that the tumor will start growing. That's the tumor challenged by a T cell having a CAR inside. There is a killing effect, but as the tumor is not totally killed because it's fairly resistant, and you can totally improve eradication of the tumor by having this spray of IL-12 right on the tumor and totally obliterate the tumor by having this enhanced effect. That's probably the beginning, I would say, of what kind of synthetic biology in T cells can be done. We currently have a series of products that are used in clinical trial currently, so UCART19, the UCART123. First patient, as I said, has been treated in 2015, and since then are still in complete remission today, and we have like more patients, or more than 20 patients that have been treated since then. Cellectis is not a provider of TALEN, and we're not a tool provider. We're more like an oncology company, and we're excited to share our technology with the Genome Project Writes. But the fact is, like, we hope to sell one day more therapeutic products, but I wish that none of you in this room will ever use our product forever. But the fact is that, like, the use of TALEN, I think, can be definitely differentiative in this type of application, because, like, the flexibility and the ability to induce like double-strand break, induce homologous recombination, gene conversion is really huge. Finally, one thing I would like to say, the synthetic biology use and application is, for example, here you can replace the track locus by CAR, the beta-2 microglobulin by NK inhibitor, and you can see that the change here, like NK cannot attack the cell, but the cell can be attacked by alloreaction of the T cell. If you replace only the track locus, then and the beta-2 microglobulin, then you open the gate for NK to attack our cells. But by blocking the cells with NK inhibitor, you can not only block the allo reaction of the T cell, but also the NK attack. And the efficiency of this is like over 40% in bulk transduction for double knock-in homologous recombination in T cells using TALEN. So today, cells are medicine are turning the page of this prehistory, and I think this will create an unprecedented revolution in human healthcare, and I'm convinced that in the coming decades, this is going totally to change lives. Thank you very much, and thank you for accepting us and contributing with this. Questions? Very nice talk, Andre. Thank you. The open question, I just want to solve it. If you're interested in talents, this is where you can call Thermo Fisher, because we will provide talents.
I know.
So when the talons go into the cell, is it some sort of a protein nanoparticle, or do you have to deliver the DNA to make the talons into the cell? And if it's the latter, then aren't you having to synthesize DNA to make a new talons?
Well, yeah, you have currently the way to synthesize a new TALEN is essentially done by Golden Gate cloning, and it takes 5 days to make a totally new TALEN, so it's pretty easy. In T cells, TALEN are transduced by, not DNA, you cannot transduce DNA in T cells because it's fairly toxic and the efficiency is pretty low. So we do this essentially by transduction of RNA, electroporation of RNA. Here you can get, so that's a real key event, because it's not that much the efficiency of transduction that is important. It's not that much the efficiency of knockout or knock-in or whatever. It's essentially how many cells you start with. For example, if you start with 1 billion cells, you transduce this one, like 90% of these 1 billion cells, and you have a 99 or 95% cell survival at the end. You might end up, if you have an 80 or 90% knockout or knock-in efficiency, you'll end up with 800 million cells. And that's very important in terms of having enough product to work on. You can raise the efficiency at the end of knock-in or knockout, but if you lose in yields or you lose in expansion of this T cell, that's pretty bad. So it's not that much a race to have a very high transduction efficiency, a very high efficiency of knock-in or knockout. Essentially, not whacking the cells too much by electroporation or by the enzyme itself, but keeping the cell pristine of any challenge and in a very good shape up to the end. That's the real challenge behind this. Now you can do profection of talin. It's a very good protein to be synthesized in vitro and to be transduced afterwards. We've done this in plants. We have like a plant subsidiary called Calyx based in Minnesota. And actually we do synthetic biology in plants. So we have like 7 plants that have been approved by the USDA. The recent one, which is high fiber wheat, a very interesting product. And the real interesting thing behind it is that if you just profect a protein, it's a mutagen. It's not transfection of nucleic acid inside and can go and make a cleavage like a chemical. So like all the concept of providing in the plant a nucleic acid makes it a GMO. Here, providing a protein, it's not really a GMO, it's like a chemical. So it's similar as everything that has been done during the 20th century. And everything that has been approved by the USDA is not considered as GM currently.
Okay, I think we're going to have to stop there, so thank you very much.
Oh, thank you.
Great presentation.